All right, chapter 3, part 4, we're going to talk about proteins. So proteins are involved in nearly every dynamic function in your body, and they are very diverse, with tens of thousands of different proteins, each with a specific structure and function in the human body. It's really important to know that structure is critical for function. So with proteins, we have 20 different amino acids. So we have 20 different monomers. All right, so here is our monomer. All right here, these would be our polymer. All right, so we have what's called a polypeptide, and then we have a polypeptide and protein can be interchangeable. And I'll explain that a little bit when we get into structures. All right, so amino acids, they have an amino group. So right here is your amino group. You have a central carbon, and then you have a carboxyl group here. This carboxyl group normally loses an H+. In solution in water, which makes the solution acidic, which is why it's called an amino acid, after the amino group and then the group that loses that proton, that H+. What is different here is this R. So what makes the 20 different monomers different is the R, or the side group. So we're just going to use the R to represent that is different. That is what is different with each of those 20 different amino acids. Here is a couple of those amino acids. We generally classify amino acids as being either hydrophobic or hydrophilic based on their side groups. So we can see here we have all of these oxygens that tend to make those side groups polar or water-loving, versus we have all these elements here that are sharing nicely with one another, so they're non-polar bonds. Amino acid monomers are linked together in a dehydration reaction. We're going to join the carboxyl group of one amino acid with the amino group of the next. That bond, it is a covalent bond, but we kind of give it, like you could say, a proper name, and we're going to call it a peptide bond. Additional amino acids can be added, have the same process to create a chain of amino acids called a polypeptide. So here, we used to call these groups, these amino acids, used to be called peptide residues, which is where we kind of get this nomenclature from. So with those peptide residues, we would pull out our water, right? It's dehydration, and then we would get a dipeptide, 2 peptide bonds, and then that bond in between there is called a peptide bond. All right, so we can see that being held together. So when we have many of these peptide residues together, we'll call it a polypeptide for many. One of the important things that proteins are in the body is they function as enzymes. An enzyme's job is to speed up the rate of a chemical reaction. So we can see here, this would be the protein. Very important to shape. The shape has to fit the substrate, what it binds to. And as it binds to that, it's going to change its shape a little bit and break the bond there and make 2 products. So this would be a catabolic. This would be a catabolic reaction. We have all kinds of other proteins in the body. We have structural, and that would be like cartilage and bone and other collagen, other things that help give our body structural support. Contractile would be muscles. Defensive could be your antibodies. Signal proteins like hormones, and we also have other types of chemical messengers called secondary messengers. We have receptors that are on the surface of cells that send messages. Transport, hemoglobin transports oxygen, and that is another example of a protein. And we have some proteins that store store amino acids or other food or nutrients for growth. A polypeptide chain contains hundreds or thousands of amino acids linked by those peptide bonds. The amino acid sequence causes the polypeptide to assume a particular shape. The shape of a protein determines that specific function. So we're going to get into this shape of proteins here with the next few slides. Now, the shape, if the shape is altered, so I have a nice folded up protein here. If I unwrap it, it will not work anymore. This process is called denaturation. So denature, it unravels, it unfolds, it loses its shape, it loses its function. I usually use the example of a paper clip. You know, if you take a paper clip and you unwind it, it won't hold papers together anymore. All right. You can try to put the paper clip back together, but it's just not going to go back the way you had it originally. And that is with many proteins. Once it's denatured, it's done. It has to be broken down. We need to make more. There are a few instances where we can refold the protein, but we're not going to get into that detail in this class. Different things that can denature a protein is salts, pH, or high heat. And this is what we do when we cook. When we cook our food, we're using heat to denature the proteins in our meat. And that makes the meat much more digestible and easier to chew. Now we're going to talk about the 4 levels of structure. The first level, so we're going to talk about primary, secondary, tertiary, and quaternary. And we can kind of see all of them are here, but I want to go through, so this whole picture here, but I'm going to go through and talk about each of those particular levels. So the first level is the primary. The primary is the order in which the amino acids are assembled. So how are we putting those 20 amino acids together? The DNA actually gives the instructions to the ribosomes. We talked about ribosomes making proteins. So the DNA is going to make a copy of itself. It's called messenger RNA. We're going to get into this process. And that messenger RNA, which is going to be a copy of the DNA's instructions, is going to go to the ribosome. And the ribosome is going to be told specifically how to put those amino acids in order and to form those peptide bonds in that specific sequence. That is the primary structure. The secondary structure results from the folding of the polypeptide. So we're going to get some interactions here. We're going to get some hydrogen bonds. And those hydrogen bonds, depending on the order that those proteins are put together, will give us one of 2 possible, actually we have 3. We can say there we have 3 structures. We have an alpha helix, which is this loop here. We have a beta pleated sheet, which is like this folding. So I always say this is like using a curling iron, right? If we use a curling iron, we get our nice little curls. If we use a crimp iron, right, we get our nice pleats. Or we have just kind of a random area where it doesn't really fold up into anything. The tertiary structure is when, even with those secondary structures, it's going to fold up on itself based on other side groups. So here we have some more hydrogen bonds with different areas, right, of our protein. We also have some amino acids that have sulfur in there so they can form these disulfide bridges. We also have some side groups that are positive and others that are negative, and so they're going to be attracted to one another. And so we have these other interactions that are going to give that protein its specific shape. Now, when we talk about proteins, all proteins will have the primary, the secondary, and the tertiary shape. Not all are going to have the quaternary. The quaternary is where, so if I go back, we talked about, let's go back. We could call this, basically this whole strand is a polypeptide, right? There's our polypeptide. Here's how we're folding our polypeptide. That's another level of folding on our polypeptide. Well, Some proteins, some functional proteins, need multiple polypeptide chains. For instance, collagen has 3 chains woven together to give us that structure. Hemoglobin has 4 polypeptides that come together to kind of snap together to make a functional protein. So some can function just as a single polypeptide, but others may need multiple polypeptides to come together, multiple components to come together to make a functional protein. So here we go. Here's our first level. That's the way we put our amino acids together. Here is our alpha helix, our beta pleated sheet. We then are folded up. We can see here is a picture. There's one of our alpha helixes. These arrows are representing a beta pleated sheet. And then, like I said, here is area that doesn't have any secondary structure. Okay, so then this is all folded up on itself, right? And so that's the tertiary. Now here is a type of protein that has 4. So we have one back here, one up here, one back here, one up here. So we have like 1, 2, 3, 4 polypeptides that come together to give us that quaternary structure. All right, so that ends my presentation on proteins. Make sure you do read your book in that section on proteins. Hopefully that will, if you read it and you didn't understand it, maybe you can go back through it again after this presentation. You do your guided reading if you find those helpful. I have that video with Mr. Anderson that if you want some extra information and extra lecture, you have it there. And do make sure work on your vocab cards. All right, I will talk to you again in our next presentation.